Hello, everyone. For today's lab, we're going to be doing a fair bit of plant observations because the goal here is to become familiar with the diversity of plants that we have on planet Earth, some of the major phylogenetic clades. So, depending on where you're at with lectures, you may have already gone over the broad plant tree of life, but I think it's appropriate to start our lab today with a quick review of how that looks. Because your first assignment in the lab is to draw the basic plant phylogenetic tree with our 6 lineages in order, those being the very simple mosses, followed by the lycophytes, those plants with scaled leaves, single vein, the ferns and their friends, or the ferns and their allies, abbreviated as fern and F, and then groups that you will be more familiar with. The cone-bearing gymnosperms, and then 2 different groups of flowering plants. Within the broader angiosperms, you have the monocots with parallel veined leaves, and the dicots with netted veined leaves. Now, the ordering on this tree is actually follows the order in which the species are listed. So we know that green land plants share ancestry with green algae. So we'll have green algae as our outgroup. And then we can just go ahead and plot each of these on an individual branch. Mosses are the earliest diverging group. These are very unique because they are gametophyte dominant. More on what that looks like later. The other thing that you can remember is that this is the only group that is non-vascular, meaning that these plants do not have veins. That keeps them from getting tall. Our next group are our lycophytes. These plants have scale leaves or microphylls. 2 terms for the same thing, meaning that their leaves have a single vein, no more. The next group is a little bit more diverse, hence why it's got a little triangle above it. It's the ferns and the allies. Now, the ferns and their allies have a number of traits that are specific to them, but most notably, this entire lineage, everything afterwards, meaning that these groups and the ferns have megaphylls, or leaves with more than one vein, something that you're much more familiar with. For the ferns themselves, you can go ahead and say that this is the last spore-bearing group. Spore-bearing with megaphylls, very characteristic of our ferns, as we'll see today by looking through the greenhouse for some diversity. Next up, we have our gymnosperms, and these are, let's go ahead and see if we can fit that in. Gymnosperms are characterized by cone-bearing structures. Of course, the other groups have flowers, the angiosperms. The angiosperms are divided into 2 different groups. Does not matter the order in which you list these, the monocots and the dicots. You can flip those 2 around just fine. The monocots have parallel veins, whereas the dicots have netted veined leaves. And we'll take a look at some of those today. And this is the way in which I want y'all to get used to drawing these phylogenetic trees. Always having some sort of root, and then each branch showing ancestry and where we have things diverging. So for example, we know that monocots and dicots share an ancestor with each other before they do with gymnosperms. But gymnosperms, monocots, and dicots share an ancestor before those three groups share ancestry with the ferns. So in that way, we show which groups are related by branching. The last component is to make sure that you are keeping track of these traits. We're going to be talking about a lot of traits today. That's the whole purpose of the lab, to kind of explore trait variation in each of these groups. So make sure that you're jotting these down. On the exam, you will be expected to know at least a few of these. But here we have our basic diversity of the plant phylogenetic tree. So now let's go ahead and wander in the greenhouse looking for our first group, the mosses. All right, everyone, so here we are in our greenhouse, obviously surrounded by a lot of pretty plants, bananas here. Today, you're exploring all the diversity that we have here, but we're going to start out with our simple plants, the non-vascular bryophytes, or, for your purposes, the mosses. Now, this sample is not living anymore, but it does have exactly what we're looking for. You notice all of the brown moss that would have been green at some point, that is the vast majority of its body, that's the gametophyte. All of the little tendrils, and can you see the fact that they're sticking up above? Those are the sporophytes. So let's go to the whiteboard really quick to talk about the genetic difference between those two groups and why it matters. Okay, so we have our log. I want to just quickly illustrate what we're looking at. We have some low-lying material that would have been green. And green obviously means photosynthetic. And then sticking above that, we seem to have these tiny little structures that might look like this. And there are lots of them. Now, the green stuff, and these are brown always. The green stuff is the gametophyte, meaning the gamete plant, the sex cell body. The brown stuff is the sporophyte, or the not reproductive body, the regular body. The sporophyte is diploid. The gametophyte is haploid. And this is totally different from the way that animals do things. You might be familiar with the animal life cycle. You have a diploid body that then will perform meiosis to make a haploid egg or sperm. The haploid egg or sperm then immediately fuses with another haploid egg or sperm to make your diploid. So you've got fertilization, mitosis as a diploid, meiosis, fertilization. Three-step process. This is different from the way that plants do it. If you want to scoot with me, if you can see this. Plants have this sporophyte generation, which is diploid. It then performs meiosis to make spores. The egg and sperm then go on to perform their own mitosis. This gametophyte body will go on to perform its own mitosis. This is similar to your egg or sperm going on to make their own body. Think about how weird that is. It's totally bizarre. It's a very different life cycle. After this has made its body, it will then go through the process of fertilization. And from fertilization, you get the sporophyte again. This fact that these plants are able to perform mitosis both as a diploid and mitosis as a haploid is what we call the alternation of generations. Because plants are making two different body types
for the same genetic individual. A body type that's haploid and a body type that's diploid. And on your mosses, the sporophyte is the brown elongated structure, and the vast majority of the body is the green low-lying gametophyte structure. That means that when you lay down on a bed of moss, you are laying down on the body of its egg or sperm. While slightly horrifying, I hope it helps you remember this very unique feature about these very cool plants. All right, so now let's take a look at our next lineage on the phylogenetic tree, our lycophytes. So when we walk in the greenhouse, we're going to be looking for things that really like the water. These are plants that really dominated planet Earth during the Paleozoic. They love swampy environments. In fact, we do have a couple of them growing in our aquarium. So we'll take a look over there towards the end of this. But our lycophytes do not dominate the Earth anymore. They are still present, so they're not completely gone. But you have to really know what you're looking for to find them. So let's take a look. So not flowering, not this guy. We're looking in here, in our terrarias. Now this is a very happy, healthy lycophyte. I'm gonna pull off a piece of it, and let's take a look here. So you see how each of the leaves are very tiny, scale leaves. You can see them very pronounced around the stem here, one at a time. They are defined by having a single vein, just one vein per leaf. They also do this, what's called dichotomous branching. This is the old school way of plant branching. They form a fork. You see a fork here, you see a fork here, you'll see a fork here. Everywhere where the leaf is branching, it always forms a fork. Let's go ahead and take a look at this under the dissecting microscope to see how it looks, so that way I can convince you of the fact that there is a single vein. So we've got the sample here, and it's in focus. So here you see that structure, you see the darkness of the vein in the leaf. Only one vein in the middle of the leaf, characteristic of our scale leaves of our lycophytes. Let's see if we get that back in focus again. There we go. Now the last lycophyte will be over here in our aquarium. Again, characteristic of the fact that it loves water. These isoides have very, very long leaves, but they still only have a single vein. All right, so 2 down, 4 to go. We've covered our mosses and our lycophytes. Mosses being non-vascular, lycophytes being vascular, but only one vein per leaf. Our next group, the ferns and their allies, are plants that have megaphylls, many veins per leaf. So let's go ahead and take a look. So here we have our fern that has these characteristic megaphylls, but we're going to start with 2 of its allies. First and foremost, Equisetum, or horsetail. This plant is unfortunately not reproducing right now, but it has spore-producing structures. Very, very characteristic of these old school plants. They snap quite easily, so you might recognize them from playing with them as a child. They have these little individual divots. Each individual divot or node makes its own tiny little megaphyll. They branch and wrap around the stem. So if you get a chance, come and take a look at these next time you're in the greenhouse. these produce spores. Another spore-producing plant that is, again, a relative of our ferns is Psilotum. And when you look at Psilotum, pay attention to these nodes at the tips, or on the branches here. These are its spore-producing structures. In a few weeks, this plant is very happy, and it will turn yellow. Now, the other thing that you should notice is that it branches with a Y. That forked branching that we see so characteristically in our early land plants that have a more simple morphology than our later land plants. So take a moment to draw both the plant architecture of Equisetum, horsetail, and Psilotum, the whisk fern. And so here we actually have some new growth of Psilotum coming up. You can see that it comes up in these little clusters. It is not yet branching, but they will be branching. I thought this plant was dead, kept it in the greenhouse for a while, kept fertilizing it, and we've got plenty of new little shoots coming up. So quite cool. We also have similar structures, rhizomatic growth from this fern. So these are kind of aerial roots that will grow along up until they find a place to grow and establish a new whole plant. Our next plant that we're going to look at, though, are the megaphylls of the fern. I've got a very particular fern here. Same species, but it's had a lot of new growth. So let's go ahead and take a look at it next to the ground. So if you look at each of these individual veins, each individual leaf, you will see that there are multiple veins. There's one main one that goes down, and then lots of individual veins along the way. So there are branching veins off of this. And this is what we're talking about when we say megaphyll. These leaves have multiple veins. And you are also asked to identify something called a fiddlehead. Well, a fully opened fern leaf is called a frond. But a leaf that's still unraveling, like this one here, is going to have its tip knotted up in a little itty-bitty ball. That ball is a fiddlehead. It makes a little spiral. And when that leaf continues to grow, the spiral will continue to unravel. So I'm going to unravel this fiddlehead here. You can see how much length there is to it. Let's see if I can peel away the back leaf here and show you that there is a spiral shape to it. Not very visible here, but that's okay. So the fiddlehead is the emerging fern frond. The fern frond is its megaphyll leaf. So here we have our next sample, a pine cone. This in particular is from a sugar pine, the biggest cones produced on planet Earth. This is just about the size of my forearm. It's fairly huge. Cones are the reproductive structures of gymnosperms. These no longer reproduce via spores. They instead make seeds. Angiosperms and gymnosperms are seed-bearing plants. Pine cones have seeds. Flowers make seeds. Our ferns, lycophytes, and mosses all reproduce via spores. So keep this in mind when we're talking about the reproductive structures of the plants that we're about to go in the greenhouse to take a look at. We, of course, have some pine saplings in the greenhouse. But the major cone-bearing plant that I want y'all to see today is our Norfolk Island pine. This here is a very unique pine species. It has very tough leaves, but they're structured slightly differently. They have a little bit of a droop to them as well, kind of feathery, almost plasticky. These were around when the dinosaurs were around. So we have our gymnosperms as having dominated the planet during that Mesozoic time period, the era of the dinosaurs. There's another plant that I want you all to Google. It's called a cycad or zamia. Those also are cone-bearing plants, even though they look like palm trees. So these are our seed-bearing lineages with the tough leaves that make them the chewing fodder for our dinosaurs, or once upon a time, that bear cones. The last group of plants that we want to talk about are our angiosperms. And our angiosperms, it's very easy to identify the veins, the multiple megaphyll veined leaves that they have. So right behind you here, we have a croton. And here you actually see the color pigmentation varying along the veins. So you've got one major vein here, branching veins here, and you can additionally see veins netting off of that. And so this is one of our netted veined plants. This is an example of a dicot. This coleus, also a dicot with its netted vein. The sambucus. See the beautiful netted veins here. All dicots. The calocarpa, netted veined as well. This coleus, still a coleus, netted veined. With its beautiful flower up top, this is actually a mint relative. In terms of our monocots, we have papyrus, the plant that ancient Egyptians made paper from. It has parallel veined leaves. So all of its veins run vertical along its stem, very similar to our grasses. Panistium, the fountain grass. You can see here, as I'm sure you're very familiar with, that all the veins are parallel on this. But those aren't the only monocots that we've got in the greenhouse. My personal favorite monocot that we've got is our Cavendish banana, a little dwarf plant sending up its new leaves. Its veins, even though they're not, even though there are many of them, you can see the beautiful banana leaf veins, they run parallel to one another after they leave the main stem. So bananas and palm trees are also monocot plants, but they have a little bit different morphology in their leaves, so it's not quite a grass, but they are monocots. So when you think about your monocots, think of grasses and bananas and palms. And when you think dicots, think everything else with netted veins. So hopefully this gives you an opportunity to have explored the greenhouse virtually. Next time you're on campus, take a look. You'll never know what you might find. See what might be blooming with our flowering plants. And we're catching the tail end of our corpse flower having flowered. So the very last of its blooms. Very hairy in here. Crazy looking. So please, make sure you go through the lab, identify the terms, and use Wikipedia if you need to refresh any other plant resources. But I want you all to be familiar with the different plant groups, the different plant classes, so that way you have a general understanding when you're walking around in nature and a better appreciation of the diversity of life on planet Earth.